Associate Professor David Beale, and he's a senior research scientist in CSIRO Land and Water, delivering research outcomes and creating impact in applications of functional systems biology, i.e. metabolomics, lipidomics, and proteomics. David is the project leader within the environmental systems biology team who develop high throughput tools for detecting, quantifying and tracking contaminants in the environment and assessing their biological impact using omic-based techniques. Welcome, Dave. You can take over the screen now.
Thanks, Brad. Thanks for inviting me to talk about some of the exciting work that we're doing in the eco-surveillance systems biology space. Hopefully everyone finds my talk interesting and informative. Happy to take questions at the end as well. Before I begin, I just wanted to touch base on some of the motivation behind this work and state the real research objectives that we're trying to achieve here in the CSIRO within this space. Simply, we're interested in investigating or how can we measure and assess environmental function, health and resilience within systems that are impacted by multiple stresses, both biotic and abiotic. In many instances, these stresses are within normal ranges of acceptance, below guideline values, or simply just not known or not quantified. And if that wasn't difficult enough, we're really interested in looking at the leakages between various levels of taxa within these systems under study, whether it be from microbes all the way up to apex predators such as sharks and crocodiles, which is some of the work that we're doing now. And in doing that, we really hope to identify a suite of tools that will facilitate this assessment across multiple ecosystems and habitats, orders of taxa. So it's not an easy challenge, but we like to sort of bite off more than we can probably chew. We think that the utility of omics-based approaches integrated as a systems biology toolbox will provide this insight and a path forward to examining these systems, but examining these systems at a scale that we normally haven't done before. So I thought it'd probably be good to sort of have this slide in there because, systems biology as a concept isn't probably one that's common amongst the emerging contaminant community. So it's probably best to take stock of what systems biology is, especially from our perspective. So systems biology as a term and a philosophy really does mean different things to different people. And the key point of difference being the consideration from a wet lab or a dry lab perspective. And by that, I mean, the people who are really in the lab generating data, processing samples and putting it through instruments to get data for people that then can take through into computational models and do that sort of data integration and data analysis offline. And this can be compounded even further depending on the lens of omics being applied, whether it be a top-down genomics approach or something bottom-up and really being driven by the phenotype or the metabolites in our instance. Our team is very much interested on those functional outputs of these systems under study. So metabolomics being the closest to the expressed phenotype, and we work closely with others to bring in that capability of the genomics and transcriptomics that we don't have inside our team. And for many of these, the perception of these omics tools is that it provides a clearer view of the system under study. That through sequencing and analysing biological function of a system under perturbation or exposed to contaminants, that suddenly we get new insight and things, or the picture just suddenly becomes a lot clearer. And to a large extent, that is true. But the reality is, the closer you get to a defined phenotype within a complex ecosystem, the noisier that omics response can be. It is a complex, busy system with competing processes and resources, and the time of sampling is also critical. And it can be the difference of trying to navigate a major motorway during peak hour or versus off-peak. So it is a complex space to be working in. Of course, there's several approaches to overcoming this omics noise. One approach is to conduct these experiments in a lab under well-controlled and well-defined conditions. And to a large extent, this is happening within the ecotox space, and the level of complexity and experimental design is increasing. But within that sublethal challenge experiment space, there is still a lot of a lot of noise. And this could be a mismatch between traditional ecotox experimental needs and the requirements of omics-based sampling. The other approach is where we're working at, and that is to actually sample more from the environment, actually embrace that noise with the aim to smooth out and harmonise a biological response. And with that in mind, so the focus of the rest of my talk is to provide an example and a snapshot of some of the work that we're doing in this space, with the particular example of PFAS contaminants and freshwater turtles. The site in question relates to an industrial site in Queensland. It has ongoing PFAS issues. The concentrations have been found to be 17,000 times higher than the National Environmental Management Plan water quality guideline for the protection of slightly and moderately disturbed aquatic ecosystems. And PFAS was also found in the majority of the biota sampled at concentrations that exceeded the dietary intake guidelines for the protection of mammalian and avian wildlife. And it was during this sampling effort of the biota that several turtles were observed within these catchments. And they were later identified as a common freshwater turtle species. And as this biota concentrations were elevated in the duckweed samples and the macroinvertebrates, it seemed a logical pathway for PFAS to enter into these turtle populations that rely on this as a food source. So we were lucky enough to obtain biological samples, blood samples, via an ongoing populations assessment permit through the local regulator. And this allowed us to look at the PFAS biomagnification through the turtle population and assess whether or not there was any impact on metabolism. Blood was collected in duplicate. One was sent for PFAS analysis at the Queensland Alliance for Environmental Health Sciences at UQ, and it was found that blood PFAS levels were significantly higher in that impacted site, and it was at levels that weren't really reported in the literature, with one exception to one study that was closely linked to a downstream water body at a major airport. The concentration of PFAS was roughly 230 odd times higher than the measured water concentrations, which clearly demonstrates a bioaccumulation and magnification of this contaminant. But obviously what that actual biological harm was on these turtles was yet to be determined. And that's where we sort of stepped in. So that duplicate blood sample we took through our omics workflow, with the intention of trying to get as much biological information from that one single pod extraction. So we took it through for central carbon metabolism, metabolites, secondary metabolites, lipid profiling, as well as untargeted proteomics. The image here sort of outlines the overall workflow, where we added a solvent to crash that protein, centrifuge, pellet it, and then put that through our proteomics workflow, while the supernatant was extracted for lipids and metabolites. And all of them were analysed through a range of LC mass spec instruments. We found a lot of biomolecules had significantly changed, either being upregulated or downregulated in response to that PFAS exposure. This related to about 30 odd central carbon metabolites, 130 odd secondary metabolites, 60 odd lipids, and about 25 proteins. These biologicals related to a range of amino acids, sugars, and carbohydrates, as well as specific lipid classes and proteins that related to lipid biological activities. So this data really does suggest that there's a connection between PFAS and alterations within a turtle TCA cycle, and this is consistent with other sort of human-based studies and animal studies, where PFAS concentrations have been strongly associated with several amino acids linked to the TCA cycle and important aspects of energy production, such as purine metabolisms, as what we found. Pathway enrichment of some of these biological data sets also pointed to lipid metabolism being significantly impacted, which isn't a surprise because PFAS does have a similar structure to some of the fatty acids used within these biological pathways. And this really did flow through to the measured proteins that really were impacting lipid transport and binding activities within these turtles. This is all preliminary data, of course. It was only on a handful of turtles, but we did put it forward as a short communication paper in Science of the Turtle Environment. And we were happy to receive on the weekend that it was accepted for publication, so that was a win for us, but it also pointed us to a larger study that needed to be designed to look at these biological impacts in a deeper manner. And that's where we're at now. So we're currently in the process of doing a larger turtle collection study where we're getting 30 turtles, a mixture of males and females, to look at the difference between the sexes and the PFAS impact of those turtle populations. We're looking at specific organ impacts, looking at brain tissues, because there's been a lot of literature around lipid metabolism and lipid accumulation of PFAS impacted taxa within brain tissues. We're looking at liver tissue, reproductive tissue, kidney tissues, heart tissues. And we're also looking at that maternal transfer of PFAS through an egg survival and incubation study, as well as looking at the PFAS translocation into hatchlings. So still a lot of work to be done, but we're excited of where these omics-based tools are taking us in terms of getting that deeper insight into the PFAS impacts. In conclusion, I said we've still got heaps of work to do. The machines are still pushing through samples related to these turtle tissue samples. So maybe next time we can get together and I can share some of the newer data and insights from some of these samples that we're analysing now. But I hope I've demonstrated that the benefits of systems biology and these tools to assessing contaminants at the biological interface. But it should be noted that, you know, for all the benefits these tools provide, their uptake and utilisation by regulators and other agencies is still lacking. It's still, in principle, a research tool used within academia, and we're hoping to change that. That may be in part because of the omics noise that I mentioned at the start. It could be because of the complexity of systems and the detailed metadata that we're just not capturing through our regular monitoring programs. But what we're proposing is not that we apply systems biology to everything, because not only will that increase the financial burden of analytical costs and sample measurements, but it's not currently achievable within a timely manner. These are really data-intensive approaches that take a lot of time to cut through and do interpretations of the data sets. One path that we're really keen to explore is this harmonisation of systems biology outputs, looking for those common trends and functions and their deviations across multiple species and systems, multiple climates and climate pressures that facilitate this transition from wet lab-based omics studies to those computational workflows for modelling and prediction experiments, where we can then take those computational outputs and do targeted, validated analysis of specific genes and proteins and metabolites that are tied to characterised system perturbations, function and health metrics. And we see this really as complementing these current monitoring programs of contaminants and providing that additional layer of biological information in terms of function and health. And it'll be very similar to going to a GP and getting a routine blood test that tells you a whole range of things about your current health, but also your health trajectory. And we see that as a really powerful tool within this contaminant space. Of course, I'd love to say that, you know, this was a one-man show and that I could do it all by myself, but it really did take a small army to apply these tools within an environmental setting. So I really do want to thank the collaborators at DES, specifically Susie Vardy and Sandra Nielsen at UQ, but also a range of other people that really facilitated this work to take place in the first instance, but also keep going in a larger study.